Good. Hello. Okay.
Couple of people have been mailing, but it's not. Right now we have only one, but
Okay. So, good morning, everyone. I welcome you all to this special occasion where we gather to pay tribute to the remarkable Dr. Lalji Singh. It is an honor for me to stand before you today to celebrate the life and achievement of a man whose contribution to science and humanity have left an indelible mark on our world. Dr. Lalji Singh was an eminent scientist, a visionary leader, a true inspiration to all those who has the privilege of knowing him or being touched by his work. He dedicated his life to the field of genetics and genomics and made groundbreaking advancements that has revolutionized our understanding of human biology and played a pivotal role in the progress of genetic research in India. In 2004, he was awarded with nation's Padma Shri in recognition of his contribution to India's science and technology. Born in a small village in Uttar Pradesh, Dr. Singh's journey to becoming a world renowned scientist is a testament to his unwavering passion for knowledge. With his exceptional intellect and relentless pursuit of scientific excellence, he rose to become the director of CCMB Hyderabad, where he led numerous groundbreaking research projects. He developed and established the DNA fingerprinting technology for forensic investigation of crime and civil disputes. One of the Dr. Singh's most significant contributions was his pioneering work in the field of DNA fingerprinting. His efforts in this area earned him the title of the father of DNA fingerprints in India. Dr. Singh founded various institutes and laboratories in India, including CTFT Hyderabad in 1995, Genome Foundation in 2004. He also played a major role in establishing the state-of-the-art laboratories, the Laboratory for the Conservation of Endangered Species, which is known as LACUNS, a part of CCMB Hyderabad, which focused on the conservation and protection of wildlife. Beyond his scientific accomplishments, Dr. Lalji Singh was a compassionate human being with a deep sense of social responsibility. He believed in the power of education and worked tirelessly to promote science and research among young minds. Today, as we reflect upon Dr. Lalji Singh's remarkable life, let us remember him not only for his scientific achievements, but also for his unwavering spirit, humility, and his passion for making difference in the life of others. He leaves behind a legacy that will continue to inspire generations of scientists, researchers, and anyone who believes in the transformative power of knowledge. With this, I invite our respected Director Madam and respected Professor P. Balaram Sir to come on the stage and pay tribute to late Dr. Lalji Singh. And I request everyone to please stand up for this precious occasion. Uh, Thank you, ma'am. Thank you, sir. Thank you, everyone. Now I request Dr. Madhuri to come on the stage and introduce about Professor P. Balaram. Thank you.
Good morning, everybody. It's an honor to welcome and introduce Professor Padmanabhan Balaram, an excellent biochemist and a former director of Indian Institute of Science. He is the recipient of the third highest Indian civilian honor of Padma Bhushan and the TWS Prize in 1994. He has been conferred recently the 2021 R. Bruce Merrifield Award by the American Peptide Society. Professor Balaram received his bachelor's degree in chemistry from Ferguson's College, Pune, master's degree from Indian Institute of Technology, Kanpur, and his PhD degree from Carnegie Mellon University. His postdoctoral work was on the erythromycin antibiotic synthesis at Harvard University, and he worked closely with Nobel laureate Robert Burns Woodward. Professor Balaram's main area of research has been the investigation of structure, conformation, and biological activity of designed and natural peptides. He has been a major contributor to the evaluation of factors influencing the folding and conformation of designed peptides. He has authored more than 400 research papers and is a fellow of Indian National Science Academy. With no further delay, please join me in welcoming our guest speaker today. Welcome, sir. We are looking forward to hearing you. Thank you.
Dr. Lalji Singh. I knew Dr. Lalji Singh for a very long time, almost at the time that he started working there.
Maybe you can keep your talk and talk.
Oh, this doesn't just squeeze me somewhere. Okay. It's like those things that you wear in convocations, no? You're always afraid of his hats falling off and you can't even speak then. I knew Dr. Lalji Singh for a very long time, almost from the time he came to CCMB. And Lalji's work was quite remarkable because LHF introduced DNA fingerprinting in the mid-1980s. And not long afterwards, Lalji had in fact introduced DNA fingerprinting into India. And it's because of his work that several laboratories in the Hyderabad area really grew. CCMB, of course, was the place where he worked, but without Lalji, there would be no CDST, there would be no Lacrons, none of these. Unfortunately, he left us rather early and rather suddenly. I'm also deeply honored to be giving this lecture in a hall named after another very close friend of mine, Maharaj Khan, who was the secretary of the Department of Biotechnology for many years. And in that capacity, he was involved in many things. But I knew him even before he was secretary of the Department of Biotechnology, and one misses him today. I reached the stage where I am somewhat reluctant to talk about the research work that I'm doing at present, especially in a lecture which is a memorial lecture. So I am going to speak somewhat generally. And what I'm going to refer on is science in the aftermath of the pandemic. Nothing had an impact on our lives as much as the pandemic. And some years later, especially those of you who are young, I think you will one day tell your children and grandchildren that you lived through the pandemic of the 21st century. Now, I'm going to tell you a little bit about the coronavirus, and in my lecture, I'll touch upon a little biology and a little chemistry. But on the slide are pictured the two foundational pillars of chemistry and biology. Mendeleev on the left and his periodic table, and Darwin on the right, and here's the natural selection. It turns out that I'd made this slide a couple of years ago in the early part of the pandemic when online lectures had become very popular. And so you were asked to give online lectures almost in every college which was there in India. You had to keep the students busy. It also kept an old retired professor like me busy because I could be online most of the time. And so I would make slides. So how do you begin? So I made the slide. Now I find I'm quite topical, because both Darwin and Mendeleev have apparently been removed from the 10th class textbook. Now here I am for the last 3 years talking about how important they are. And here is the government now, NCRT no less, removing them from the textbook. So let's see what happens as we go along. Are Darwin and Mendeleev really important? Now, very many times when I'm asked to speak, especially to general audiences, suddenly someone asks you a very difficult question, they ask you what is science. At the moment, at the National Center for Biological Sciences, where I sit as a freelancer, it turns out that we have an Obaid Siddiqui visiting professor there who's a linguist. He's a very famous linguist in India called G.N. Devi. And what G.N. Devi does is he conducts these meetings. And if you go and attend the meeting will be a discussion meeting on asking scientists, what is science? So suppose you're suddenly confronted by someone asking you, what is science? Well, you don't know what to say. Because science is what I do and what other guys whom I know or what they're doing. The science, actually, probably the best definition of it, is that science is the study of nature. And now, the moment you say science is study of nature, then someone asks you, what's the definition of nature? What do you mean by nature? I looked for a definition of nature, and the best definition you will find is in volume 1, number 1, of the journal called Nature, 1869. So the first issue had an editorial. Why are we having this journal? How many of you want to publish in the journal Nature? Wonderful. Everybody put up their hands. I'll put up both my hands. And I thought this is something that we should pay attention to. Now, Thomas Huxley, the famous biologist, was asked to write this editorial. He didn't write the editorial. Instead, what he did was he translated from the German an essay written in the 18th century by the German poet von Goethe. And what Goethe said was this, and I paraphrase it: nature, we are surrounded and embraced by her, powerless to separate ourselves from her, and powerless to penetrate beyond her. You can't get a better definition of nature. And it, of course, turns out that the coronavirus was a force of nature. And nature is remarkable. Nature is so economical. Today, everybody is talking about energy. Everybody's government animals. They're going to spend 20,000 crores over the next couple of years in doing hydrogen energy. Now, you can ask the question, what is life? Life, of course, is all about energy. You have to produce energy in order for an organism to live. And our organisms do this. We, of course, produce energy in our mitochondria. We do this by oxidizing substrates. And in the process of oxidizing substrates, we also couple it to the phosphorylation of ATP to DATP. That's oxidative phosphorylation. But in this process, what happens is that all the electrons that they're stripping off our substrates have to go somewhere. Those electrons now get into oxygen, and we convert oxygen into water in cytochrome oxidase. Go back and look at oxidative phosphorylation. Very, very important. Old stuff. On the other hand, there's only one source of perpetual energy, at least perpetual energy for or maybe a time scale much longer than any of us would be worried about. That's the life of the sun. So although there will be astrophysicists now telling you that the sun will eventually run out of energy and die, but that's a long way into the future, so we won't worry about it at the moment. It's photosynthesis. Photosynthesis is the only way to harvest light energy. Now look at what happens in photosynthesis. In photosynthesis, exactly at the end of it, after you've cooked carbon and made carbohydrate from it, water gets into oxygen. So it's exactly the best. So what photosynthesis has now done is it has split water. But when it has split water, it hasn't split water into oxygen and hydrogen. We would like the hydrogen, but it hasn't split water into oxygen and hydrogen. You go back and think about it a little bit. It's taken away the reducing power. It's taken away the electrons, and now it's made things which the cell needs. It makes molecules which now contain energy and reductants, like NADH and so on, which are now produced for other metabolic purposes. But in this cycle between plants and animals, you're an institute which deals with animals. You're a great believer in institutions which don't separate any subject from any other subject, because every subject is interconnected. And it turns out that plants and animals now are intimately interconnected by the global carbon cycle. Now, the only problem that we have today when we talk about climate change and so on, we are putting out more carbon into the atmosphere by our own other activities and activities which need to be controlled to some extent. So what about Mendeleev? Look at Mendeleev's periodic table. I recently found a periodic table which is called the medical periodic table, which looks at all the elements which are involved in biology and medicine across the periodic table. Very likely. But most interestingly, look at carbon and silicon. You need carbon for biology and life. Absolutely essential. There's no organic chemistry without carbon, and there's no life without organic chemistry. For the rest of it, all of this, that which we can't survive, requires the next element in the periodic table, that is silicon. So if one knows even a little bit of chemistry, one might ask, can silicon now replace carbon in all living processes? And you might think about it and ask the question, why silicon cannot do this? But at the same time, silicon is very useful to us. Without silicon, the digital age would not have been born. So they are connected. Where does this carbon come from? This carbon, of course, comes from the stars. That's the only place where you can make the elements. And if you haven't read this book, go back and get hold of it and read it. It's an old book, 50 years now, and this is why I think it's worth putting it up. This is based on a BBC series called The Ascent of Man, narrated by the theoretical physicist Jacob Bronowski. And it appeared then as a book. And what Bronowski says here is, in all the stars, they're going on processes which build up the atoms one by one into more complex structures. Matter itself evolves. The word comes from Darwin and biology, but it is the word that came to physics in my lifetime. Keep the word evolved very much in your mind, because in all the discussions that we have today, many of them extremely contentious, the word evolved should resonate, because evolution happens not only in biology, but evolution happens also at the level of human cultures. They have evolved over the years. He talks about the formation of carbon, and you must see where the carbons come from, all the carbon that's on Earth. It's formed when two nuclei, three helium nuclei, collide at one spot. Actually, transiently, they give you beryllium, and then a third one comes along and hits it, then you get carbon. This, he says, happens in about a millionth of a second, 1 billionth of a second, 10 to the minus 12. Every carbon atom in every living creature has been formed by such a wildly improbable collision. So this should teach you that life itself is extraordinarily improbable, very improbable, but still we're all here in such large measure. So if you're a biologist, you must actually ask this question, which you never ask. I ask what is science, but you might ask more fundamental question, what is life? And sometimes your whys are not thoughtful. But if you do ask these questions as you get older, then you have to start reading. And as you start reading, you find that you have to go to subjects which you do not really know. And you have to read about those and eventually try and connect them. I found this picture here in an article in Science Advances in 2017, which was called, the figure was called the timetable of evolution. Now, everything that's written there in yellow and highlighted in yellow is put by me on top of the author figure. This tells you how the disciplines of science had sort of evolved, what we study as science, how they had evolved over the years that the Earth has been around. The Earth has been around now, and this is well estimated, for about 4.6 billion years. And now, if you look at 4.6 billion years, you will find the first subjects that one needs to study are geology and chemistry, because the Earth inherited everything. Most important thing about Mendeleev's periodic table is this, that all the natural elements which are there in Mendeleev's periodic table were the Earth's inheritance when it was born. There's no more of it. So if you want more lithium for your batteries, you have to mine it. So without digging and doing environmental degradation, you're not going to get any of the elements that you need to use. Whether it's coal for energy or it's lithium for your batteries or anything else. So geology and chemistry sort of go along. Then one might ask, when did biochemistry evolve? So biochemistry must have evolved, to some extent at least, before the first cell evolved. And so when did all of this happen? You might turn out that you will get an idea that somewhere around this period here, you begin to get cycles, some of the metabolic cycles, and some of the early cells, archaea and eubacteria, both of them would have come over here. And about 2.5 billion years ago is when photosynthesis evolved. The first photosynthetic organisms were, of course, microscopic organisms, cyanobacteria. And without photosynthesis, there is no oxygen in the atmosphere. And, but we need oxygen. We can't get by without oxygen. So before that, anaerobic life existed on the planet. And then with the sudden evolution of photosynthesis, the Earth becomes oxygenated. And as the atmosphere becomes oxygenated, more things happen. Somewhere around here, maybe just before oxygen, is the first appearance, and people don't know this very well, of the first eukaryotic cell. You might ask the question, how did the first eukaryotic cell actually evolve? Because it's only after eukaryotic cell evolved that biology really boomed. So up to here, you can see we've come to biochemistry, and then we've come to microbiology. Now we have a rather long lag period when things are going on, things are getting better and better. Animals and zoology appear much later. And something that should surprise you is bacteria and plants appear even later. So in many respects, plants are quite complex organisms, and they have many, many capabilities which the rest of us animals don't really have. Medicine, which we all would like to do, especially if you're a student first graduating from college, you want to go to school, you want to go to medical college, medicine is very recent. It's only after the evolution of human beings that we really worry about medicine. So this transition of human beings from animals to what we now consider as human is fairly recent, and maybe in less than the last 500,000 years, maybe even less than that. Now let's get to the virus, because we have to think about the virus. That's what, I don't know what it was with all of you, but I remember in March, towards the end of March of 2020, suddenly one day, the Prime Minister came on TV. And the Prime Minister came on TV and gave one of his very compelling speeches. And I still remember this. He said, I want all of you to stay at home. And then at the front of your door is the Lakshman Rekha, which you cannot cross. So we were all locked in. And once we were all locked in, the question was, what do you do? I don't know what you did, but you have no alternative. Either you look at your phone or you look at your computer. If you're young, you look at your phone. If you're old and your eyesight is showing, you look at the large screen on the computer. And that's what I do. Of course, the coronavirus pandemic, you need to reflect on it. You've very rapidly forgotten it, I realize. Is absolutely devastating in terms of the loss of life. When I made this slide sometime toward the end of 2022, the number of deaths in the world had already crossed 6.6 million. So it's probably easily the number of deaths in the world, probably another estimate must be at least 10 million. That's a huge number. Now, we had many ways of eliminating the coronavirus. There were some very interesting ones which appeared when Donald Trump decided that we could put disinfectant into people's lungs. We were not far behind. Patanjali had something called Coronel. And I rather like this cartoon which I saw, where this man says, I read that drinking beer can cure COVID and bring world peace. And then he adds, probably fake news, but why take a chance? So he continues to drink his beer. So I think science was roughly in this stage during the coronavirus pandemic. I think one needs to reflect on all the science which was done by scientists and their institutions during the coronavirus pandemic and ask the question, how much of it was legitimate science, how much of it was fraudulent science, and how much of it was simply nonsensical science. And there's plenty of it all over the literature. It doesn't matter. Coronavirus captured everybody's imagination. You know, I found all these pictures. I had nothing to do, so I put them onto a slide. You know, whether you were, that was actually a police in Secunderabad. You see somebody going on the streets, tell them to go back inside their houses. In Chennai, we had imaginative auto rickshaw drivers who attracted, I think, passengers by pretending to be the coronavirus. But I think somewhat more imaginative people in Italy and France, which were very heavily affected in the early stages of the pandemic, at least got a sense of humor. They made Easter eggs and cakes out, which looks now like the coronavirus. Because what is a virus? So I looked up what a virus is. See, what happened when the coronavirus came was we had the telephone. So I found, just as a measure, I called some of my friends who should know better because they are virologists. And it turned out that even virologists knew very little about it. Nobody heard of the coronavirus until it came. Very few people. It's only when people started dying that everyone paid some attention. The best definition of a virus that you can get in the literature, which I found with my searches, was in this philosophical dictionary of biology called Aristotle to Zoos, written by the English immunologist Peter Medawar. Medawar says that a virus is a piece of bad news wrapped up in protein. This is actually quite interesting, you know. Yeah, the bad news is the nucleic acid. And I've always been interested in proteins and try to disparage all my colleagues who work on nucleic acids. So here this is, the nucleic acid is bad news, that's what the virus contains. And transfers it into you, and then more viruses appear. Of course, with the coronavirus, not only is there protein, but there's also a phospholipid bilayer. And it's an enveloped virus. And if you look at it, that's what it looks like. These pictures are old pictures. I'll come back to them in a moment. But more than a century ago, William Osler, who was the founding father of modern medicine, he actually said soap and water and common sense are the best disinfectants. This is why we're washing our hands and putting organic sanitizers all over there so that our lipid bilayer now dissolves away, the virus disintegrates, and we're all safe. Didn't quite work that way. Everybody finally got infected one way or the other. What was abundantly missing during the pandemic, soap and water were there. Common sense generally was missing. But also it turns out that common sense is actually very uncommon and usually not there. So at this point in time, with nothing else to do, I decided that I must study about the coronavirus. And my advisor who said I should study about the coronavirus was none other than Al Pacino acting as Michael Corleone in this Godfather 2. Now he says, keep your friends close, but your enemies closer. And already Mr. Modi had, by that time, invoked images of the Mahabharata war, the number of days that it would take for us to overcome the coronavirus and so on. And therefore, coronavirus was a declared public enemy. And so we have to study the coronavirus. So I did study the coronavirus. I did publish, and the journal where I published, or I show you here, it doesn't have an impact. Now, what was I trying to do? Since none of my friends seemed to know what the coronavirus was, I asked a rather simple question. When was the coronavirus first discovered in the scientific literature? When did the coronavirus actually appear? So around that time, the BBC and other places pictured the lady here on the right, June Almeida, and said she was the discoverer of the coronavirus because she took the electron micrographs that you see in the center over here, these micrographs. You can see the corona, see where it gets its name. And she worked with the English virologist, David Tyrrell. This is one of the very early electron micrographs of a virus. And David Tyrrell was a very prominent virologist working on the rhinovirus, which causes the common cold. It turns out that about 30% of the common colds that we've always been getting are coronavirus colds. And Almeida and Tyrrell published this paper at that time. Now they said here, and this you must see, what I've marked in red, they said that the most interesting finding was 2 human respiratory viruses, and they gave these names. These are usually just abbreviations which the scientist has given, 229E and B, that they are morphologically identical with an avian infectious bronchitis virus. It turned out that the only people at that time who were really interested in coronaviruses before the pandemic were veterinarians, because it's fairly common in animals. Pigs, for example, get the coronavirus, and porcine virus causes diarrhea. Now, let us go back. Yeah. Now, who discovered the coronavirus? When you read a paper, it's very important to read the legends to the figures and everything carefully. The only thing in today's papers that you should not read are the introduction and the conclusion. If you leave the introduction and the conclusion out, because most of them are heavily overhyped and overstated, if you read the rest of it, you will get something out of it. And the experimental sections are particularly there. And there you see strain 229E is of somebody else in 1966. So they must have discovered the coronavirus because they had that 229E virus. So I went back and looked at the acknowledgements then and found they actually acknowledged Dr. D. Hamre for the 229E virus. So now I decided that Dr. D. Hamre was in fact the discoverer of the coronavirus, of the human pathogen. Fine. So went back to the literature and found this paper in the Proceedings of Society of Experimental Biology. You can imagine into the apartment thanks to the lockdown and nothing to do, staring at the computer. We had the whole internet available to search. So I found this paper very quickly, 1966, and there it is. Now I found that she says here that this is the first coronavirus demonstrated as a human pathogen. She's done everything here. I now knew it was a lady because there's the name Dorothy. It's of course a lady's thing. So now I was quite interested. There were very few women in American science in the 1960s. And therefore, here was a woman biologist, discoverer of the coronavirus, which is now creating havoc all over the world, and yet nobody knows her name. She actually found that this was an RNA virus by using DNA synthesis inhibitors. And then she also determined the size of the virus by classical methods, 89 millimicrons, 89 nanometers in present day units. If you go back to the David Tyrrell paper 1 year later, you find they've also made now from microscopy the measurements of 80 to 100 nanometers, and she's spot on. I thought this was marvelous. And then I searched further and found that in the literature, this is around when the coronavirus first appeared in Nature. So 1968 is the first use of the term coronavirus in the literature. And of course, when I knew that people start asking me to give talks, so one thing that I'd like to do in a talk is to put a picture of the person I'm talking about. So what you do now, you go and type out, okay, I'm going to Google, put images, and there I found all these pictures of nice young ladies. None of them look like a biologist. If I searched a little further, none of them seemed to fit the dates, and therefore I could not find a picture of the discovery of the coronavirus. So once I couldn't find a picture of the discovery of the coronavirus, I did the next thing that scientists, searching Google images is somewhat nicer than searching PubMed and Google for scientific papers. But once in a while you have to do that. And then I established that she was a very prolific scientist, 40 publications in PubMed between 1943 and 1972. So she went back all the way to 1946. So by this time, I had learned how to search all these things. So I even found where she was born, where she died, where she lived, because American records are available on the internet. And I found that she got a PhD in the 1930s. And very few women in America doing their PhD in the 1930s. So I thought this was even more interesting. She worked on penicillin during World War II, and she went into science during the years of the Great Depression. So I thought it was a very interesting story. And I searched further. Now, of course, my problem is to find a picture. So I finally found a reference to her, actually not to her, but to the person whom I later discovered was her husband, but her name was also there somewhere, in an archive in Arizona. So almost immediately, I was doing this day and night. So you can see 29th of April, 2020. 29th of April, 2020, right during the first major lockdown. So I shot off a letter to the curator of that museum, of the archives, asking them, see, you have this, I think she found the coronavirus and so on. And I was really surprised. I sent the mail around midnight, and early morning I get up and I find the response. They were very interested. And they said, lockdown, you can't go to the archives. Then I think they dodged the lockdown and went to the archives. Sure enough, they found photographs of her. All these photographs had been deposited. They never looked at the photograph. They didn't know who she was. And her husband had deposited them. It turned out she was a photographer. But they also deposited lots of pictures of the American Southwest. And she was an extraordinarily good photographer. So they now say that her photography matches the best that was there in the 1950s and 1960s. So I finally found her. And in my helper was really my collaborator, was this young lady at the archives whom I had never seen, but now I've got a picture of her. And she was also quite interested as to why is a crazy old guy somewhere far away across the world interested in searching for a picture like this. So eventually what happened was I wrote up what I wanted. She wrote up what she wanted in an archive magazine. And she also interviewed me for her article. Now I found that the graphs were here. And eventually, I probably had at the end of her life, went to the small town called Ure in Colorado, died. So I managed to get in touch with the people in Ure in Colorado to tell them with the problem. Eventually, I found that there's a population of 1,000 It's one of my ambitions to go to a place which has a population of 1,000. But I probably find more than 1,000 people on this campus itself. He died there. We even got a picture of a house and all, and there it is. You can see right there in the middle. So the people in this town got very interested, and they said, we have a magazine called The Historian. And this is the publication I am proudest of in my entire career. My entire article in Frontline was reproduced in the Historian in the city. So I was interviewed after this, and they asked me, why were you doing all of this? My only answer to that was, I was just curious. I had nothing else to do. And I would have only been a problem for my wife or anybody else who happened in the house if I hadn't sort of sat in front of that. And so I told her at that time that it is a tribute to the power of the internet, that the road from Ure to Colorado, which you can see there on the Google map, passes through Bangalore. It also seems to me that in some strange way, the virus has led us to discover by turning the world upside down. Because if we didn't have the pandemic, I wouldn't have done all of this. And if we didn't have the lockdown, I would have never ever thought about the coronavirus. So today, if you had a textbook of biology, the discoverers of the coronavirus are these three people here, 66, 67, and that's when the human pathogen was found. Now, why was the human pathogen not investigated? Tyrrell, who was a meticulous virologist, actually wrote this review in 1996 in a textbook of medical microbiology. What he said is coronaviruses cause acute, mild, upper respiratory infection, common cold. Once he said that, they are no longer the subject of investigation. Now, DBT isn't going to give you any money to study a virus that causes common cold. It's not translational enough. Because you don't want to cure the common cold, it cures itself. But in 2002, 2003, the first outbreak of SARS-CoV-1 happened. This happened in Vietnam. There was an SOS from Hanoi to the WHO for sending a specialist in. This gentleman, young at that time, Carlo Urbani, went in to Hanoi, and he immediately realized that this was a terribly infectious disease. Extraordinary quarantine measures were imposed, and the virus didn't spread. Urbani himself contracted the disease and died a few days later, very, very fast. Remarkable man, and because of him, SARS-CoV-1 did not actually get out too much. It was pretty infectious. But a lot of research in the years between 2003 and 2013-14 was really done with SARS-CoV-1. And it turns out that except for some details, SARS-CoV-1 and SARS-CoV-2 viruses are very, very similar. I'll come back to the virus again, but I'll digress a little bit together. Now, you need chemistry if you're going to understand viruses and almost everything in biology in molecular detail. And no one has emphasized the importance of chemistry better than Arthur Thunberg, who wrote 20 years ago that chemistry is the lingua franca of the medical and biological sciences. So unless you know your chemistry reasonably well, you're not going to be able to understand many of the things in the kind of molecular detail that one wants to today. How do I... it will move. Moves a little bit slower. Okay. The length and time scales are the only things on which chemistry and biology differ. So it's important to worry about scale. Whether you start with a simple molecule like ethane on one side, or go to something complex like an erythrocyte on the other, these are the kind of length scales and time scales that you move over. The virus you can see there sits right between the ribosomes and mitochondria. That's about the level of complexity that one has. Now, is chemistry really important? It is, because if you look at this, and what is the most important thing in chemistry? In the year 2000, many the journal Nature asked many commentators to write a one-page essay on what they thought was the most important scientific advance of the 20th century. Now, if you were asked to write what was the most important scientific advance of the 20th century, if you are a physicist, you might say the most important advance was the development of quantum mechanics. If you were a biologist, you would say the double helical structure of DNA really determined in the 20th century. But a Canadian commentator, Václav, wrote this essay, which has remained in my mind, called Detonator of the Population Explosion. And he said the world might be better off without Microsoft and CNN, and neither nuclear reactors or space shuttles are critical to human well-being. But the world's population would not have grown from 1.6 billion to today's 6 billion. Now it's well over 7 billion without the Haber-Bosch process. Now, what is the Haber-Bosch process? All of you would have learned it in 12th class, because while the government might eliminate Darwin and Mendeleev, they're unlikely to eliminate the Haber-Bosch process, because it doesn't ring a bell in anybody's mind. It is the synthesis of ammonia. The industrial synthesis of ammonia, terrible synthesis, high pressure, high temperature, catalyst, this is where you learn mass action, the catalyst principle, and so on. Everything that you learn with the Haber-Bosch process, you would want to forget as quickly as possible. And important, because without industrial ammonia, there would be no industrial urea. And without industrial urea, there would be no fertilizer. And if there was no fertilizer, then the first agricultural revolution of the 20th century would not have happened. The second agricultural revolution of the 20th century, the Green Revolution, is a revolution which came from biology. The first one came from chemistry. Biology, of course, makes ammonia much better, and one is still trying to learn whether nitrogenases can actually be mimicked. If you look at the cell, that's the cell. How many of you have seen this? Everybody. Anybody gone here? What is it? Nobody. Today on the internet, you can have all the Zoom things. You zoom into a different thing. Once in a while, you must go. It will tell you how many molecules there are. It tells you that biochemistry is a chemical nightmare. And without biochemistry, there's no biology. So there we are, living in this nightmare. And now what do we do? People try to simplify it. This is a reference I've given you, 2018, very recent, where one is trying to understand the logic of metabolism. How has metabolism evolved like this? Here, for example, are three of the cycles, two cycles and the major metabolic things. Glycolysis, the citric acid cycle, and the mitochondrial oxidative phosphorylation complex, all coupled to one another. And this evolution of cellular metabolism is a real wonder. Without this, no living cell. Now, of course, you might also ask the question, how do you generate energy? It turns out that most of the time, we do it with molecules, chemical energy, ATP, and so on. Now, you see phosphates all over the place. Now, for example, Chris Walsh and his collaborators identify 8 kinetically stable but thermodynamically activated molecules that power cellular metabolism. What does this mean? Kinetically stable means these molecules are intrinsically unstable, but they are prevented from activity by a high barrier which prevents them to go to their products. Therefore, they're kinetically stable, but they're thermodynamically activated already. They can give you energy. And this is a very interesting way of actually looking at cellular metabolism. I've shown you a little bit of the complexity, so we can return to the virus. See the paper, 2006, already electron cryomicroscopy. SARS-CoV-2 is still in the future. And you can see, there it is, mean particle diameter now with the latest techniques, 82 to 94 nanometers. And Dorothy Hamby's measurement of 89 nanometers falls right in the middle. This really made me feel very, very impressed when I saw it. Amazing termination. Today we know all these structures. Cryo-electron microscopy now allows you this determination. They were all done for SARS-CoV-1, so they were immediately done again for SARS-CoV-2 very quickly. The enormous scientific advance that has taken place over the last 3 years is because of all the work that was done in the intervening decade between COVID-1 and COVID-2. We've got receptor complexes and so on. So we get all these structures in great detail and what atomic level detail. And therefore, even if you're a biologist, you must ask, what's the use of these atomic level details for all these complex structures? Millions of atoms connected to one another. Now, atoms are terribly important because the importance of atoms can't be better illustrated than by going back to the school of physicist Richard Feynman. You go to the lectures and lectures, the immortal lectures given at Caltech to undergraduate physicists. Volume 1, chapter 1. This is what Feynman starts. He says, if in some cataclysm all of scientific knowledge were to be destroyed, and only one sentence passed on to the next generation, which is what statement would contain the most information in the fewest words? The cataclysm that he's thinking of is nuclear war. And this is the 1960s. And today, of course, if you see every now and then, Vladimir Putin will threaten us with the specter of nuclear war once again. So you might also begin to think about it. So then he says, he answers his question. He says, I believe it is the atomic hypothesis or the atomic fact. He said, if you do this, you can build all of science once all over again. And from Feynman, this is probably something that one should think about. After a digression into chemistry, we should go back to biology. If you want to digress into biology, better start with Darwin. And go back to the origin of species. And you know, Darwin says this beautifully. And especially in an institute for animal biotechnology, somehow I don't like the word animal biotechnology. It means like you're interfering with animals with something. Nature has interfered with animals by mutation and random selection, random mutation and natural selection over a long period of time and created. Darwin says this. He says there's grandeur in this view of life. And he goes on to say that while this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. And this is happening all the time. And the human being might just be a minor aberration in the grand scheme of things. There are 3 pillars of modern biology. And those 3 pillars of modern biology, of course, Darwin, more or less his contemporary Mendel, because he tells you the 2 things. One is the inheritance, characteristic passed through generations, but not identity with variation. And how are they connected? How do you connect Mendel and Darwin? The only way to connect Mendel and Darwin is to go to chemistry, and that's the chemistry of DNA. It's actually the Watson-Crick structure. So I put here Oswald Avery, who discovered DNA, and the Watson-Crick double helix, base pairs, complementarity, all of that, the information flow in biology, all of that will become here. Now you can see Darwinian selection will also happen at the level of molecules, exactly as it happens at the level of organisms. And in paying a tribute to Dr. Lalji saying, one must say all things that have happened before 2000, we had the human genome. Now we've got lots of eukaryotic genomes. This is an old tree now, then, more organisms. But what I wanted to tell you is look at this branch of the tree. Human beings, chimpanzees, rats, mice, pigs, cattle, sheep, horses, and dogs, all are just nearby branches on a very complex tree. This tree teaches you a little bit about the unity of biology. The many arguments that we will have about pigs and cattle and so on are actually meaningless in the context. Because in the context of biology, everything is very closely related through their DNA. But at the level of proteins, sequences vary by random mutation. Random mutation in the nucleic acid, random mutations in proteins, and then selection. This has happened in the case of the SARS-CoV-2 spike protein. The spike protein is the protein which projects out of the virus, the one which makes contact with the cell, with the ACE2 receptor, and then allows the virus to get into the cell. How do you understand this? How do proteins change, vary over time, over the course of evolution? Go back and read this paper in 1970 by the evolutionist John Maynard Smith. It's all of one page in data. It has no equations. It has no chemical structure. He explains what is called the concept of protein space. The concept protein space and mutation and section, he explains with this example. I put this wherever I have students in the audience because I want them to produce another example and then send it to me so that I can now claim it's my own. He starts with the word word, makes one mutation, becomes go, makes another mutation, becomes go, makes another mutation, gone, and then another mutation, then becomes gene. Remarkable. Every word now has a meaning in the English language. And because it has a meaning in the English language, what he says is, this is how proteins also work. You make mutations. If a mutation has a function, it is retained. If the mutation is nonsensical, it's discarded. And so over a period of time, beginning with maybe an ancestral protein, you have now diversified in a large number of sequences in which you have selected out for the phenotype. Now, people have thought about this over the years. Francis Arnold, who got the Nobel Prize for doing evolution in the test tube, bacterial evolution, actually has written this. She calls it the Library of Maynard Smith and my search for meaning in the protein universe. How many sequences are there? 20 amino acids written any length, in any order, whatever you want. It's infinite. It's an infinite library. And the infinite library of sequences metaphor that she uses is this little short story called The Library of Babel, written by the Argentinian author Jorge Luis Borges. What Borges talks about is a library which contains all the information that they look, and somewhere there is buried the secret of the universe. In this library, if you keep searching, that's how you will end up, because you will never ever find the answer. So sometimes the protein universe also looks like that, a very complex, large universe. But the SARS-CoV-2 protein itself has 1,273 residues. Number of possible sequence space is 20:1273, which is an impossibly large number. It's much larger than the number of atoms in the universe. So you can't think about these numbers. Even sometimes these numbers are so large that even the most imaginative scamsters in India can't think of these numbers in terms of money. It's an impossible art. Mutations have accumulated during the pandemic on this protein. And what have those mutations done? They affect these processes, binding to receptor, membrane fusion, and virus internalization. You can understand a number of things by studying these. So this is kind of work that I was doing during the pandemic. To generate figures like these, I won't worry too much about it. The pandemic was driven by a single mutation. That mutation, which is purple, aspartic acid 614 becoming glycine D614G, one residue of the 1273. So the virus which emerged from Wuhan changed one amino acid and afterwards swept through the world. And as it swept through the world, it acquired, of course, more mutations because it was faster and faster. Some of them were terrible, like the alpha and the delta coronaviruses, which appeared in between. Now, of course, we also have the more benign Omicron. So you're probably still getting coronavirus infections, but nobody's worried about it. The moment you stop, I think we all now follow Donald Trump's original injunction: don't test. If you don't test, you won't have disease. But how has science changed? Science has changed enormously during the pandemic. This is the Omicron variant. Remember, by now, I had graduated Instead of finding the picture of the discoverer of the coronavirus, I had gone to looking at sequences from the database. I couldn't look at sequences from Indian databases because they were then not allowed and they were not accessible. Ironically, it turns out that as the pandemic was coming to conclusion, I was appointed as the chairman of the committee for the National Data Set. After that, the sequences became available, but the pandemic also disappeared. But the US data was all available on the NCBI data site you could look at. It really doesn't matter. The Omicron variant appeared in November 23rd, 2021. Where did it appear? Not in a high impact kernel. The mutations were announced. And someone sent me a message saying that, Twitter, there's an announcement there. So I looked at it, I copied down all those mutations down. And then 2 days later, another Twitter which corrected an arginine to life, which I had plenty of. Also not coming so fast on Twitter. And I don't know, I'm not on Twitter, and I'm not very comfortable using it. But it doesn't matter. But that year, November 2021, February 27th of December, a paper was submitted to Science, structure of the Omicron mutant, and this structure, the Omicron, with bound to receptor and an antibody. And this structure was published, accepted, and published online on 8th of February 2022. So now you can see 12 weeks, 8 weeks, 6 weeks. This is the time scale for doing these experiments. Now, those of you who are young and positive here, really reflect the pace at which certain kinds of science can be done today. It makes many efforts sort of meaningless. This paper has just come online, 7th of June. To tell you that despite advancing age and decreasing pandemic scare, I still continue to diligently look at the literature. This paper is a very interesting one. It's caught the protein in the post-fusion conformation. Therefore, it gives a role for the most conserved part of the sequence, which is the single transmembrane domain in this protein. We're coming to the end. With all this pandemic and everything that's happened. We've had other sort of epidemics before, not quite a pandemic in the sense of SARS-CoV-2 or COVID-19, but when the AIDS scare broke out through the world in the late 1980s, Joshua Lederberg, one of the high priests of molecular biology in the 20th century, and a very thoughtful person, wrote this essay called Medical Science, Infectious Disease, and the Unity of Humankind. And then he says here in this article published in the Journal of the American Medical Association that human intelligence, culture, and technology have left all other plant and animal species out of the competition. We also may legislate human behavior, but we have too many illusions that we can, by writ, govern the remaining vital kingdoms. The microbes that remain are competitors of last resort for dominion of the planet. The bacteria and viruses know nothing of natural sovereignties. In that natural evolutionary competition, there is no guarantee that we will find ourselves. This is something that one must think about. You cannot actually get rid of microbes the way you imagine that. What are the lessons from the pandemic? Lessons from the pandemic are that nature periodically provides a reminder of the limits of human action. A number of world leaders who declared that the pandemic was over, that we have won, do not realize that what has really happened is that we've been the subjects of a grand evolutionary experiment. And in this evolutionary experiment, we always do experiments in biology on animal models. And who are the animals in this case? The only example that I know of a biological experiment where the animals are now busy looking at the results of the experiment. So nature also demonstrates that the frontiers of science are truly endless, and that biology, assisted by the chemistry of molecular evolution, is a formidable force. And were created in the laboratory by the Chinese. Was it a Chinese virus, as Trump says, or did it just come from bats and an intermediate animal, a zoonotic transfer, which all the virologists are very fond of? And we don't know. And American intelligence, which didn't prove tremendously successful in Afghanistan, and American intelligence, which doesn't seem to be proving particularly efficient in Ukraine, is still divided on the issue. Or some think it was created, some think it evolved. Now, I found this poem, I read it in school a long time ago. William Blake wrote a poem on the lamb and he wrote a poem on the tiger. And, both the lamb and the tiger are wonderfully, they have bilateral symmetry, characteristic of animals. And so they're very symmetric. On the other hand, you look at the lamb, it looks nice. You look at the tiger, yeah, it looks nice, but from a long distance, especially if you have various things separating you from it. So he asks the question, what immortal hand or eye could frame that fearful symmetry? Did he smile his work to see? Did he who made the lamb make thee? That is a question that we're asking with the coronavirus. Beautifully symmetric. At the same time, the picture evokes fear of the COVID-19 pandemic. And we do still know where he made the coronavirus. Nature or virus is living organism. How many of you would like to live a living organism? Or we should exclude them from biology. So tomorrow you might be in the position of having to be on a committee to write the NCERT textbook, and you'll have to decide whether to put the virus into the chemistry book or whether to put the virus into the biology book. How will you do it? I'm just interested. You're all animal biotechnologists, so how about for biology? Oh, minority, no, but I think they're not the I would know. Okay, so I was for chemistry. Even fewer. By itself, the virus is absolutely inanimate. A collection of molecules. Sometimes a collection of molecules is even beautifully crystalline. In the rhinovirus, for example, the crystal structure of the rhinovirus has actually been done. So crystalline. But at the same time, all it needs is then when you put into a cell, it springs to life and makes more viruses. So if reproduction is the only characteristic of life, and maybe you might put that, people are still arguing it. Does it have a place in the history of life? But it turns out that if you look at the human genome, you can find traces of viral genes everywhere. This is our past encounters with viruses over tens of thousands of years. They are embedded now into our genome. There's a lot of history in your genome. Now, because you look at this tree, you might ask the question, what was the last universal phenomenon? You have bacteria here, they obviously came first. Then you have the eukaryote, and somewhere there must be a root. And how did the eukaryote actually form? Now, it turns out today, of course, there are good theories right from the 1960s. I'm going to show them. The endosymbiotic theories for eukaryotic origin, people are looking at them quite extensively once again. What happened? And for the students, one piece of advice. If you were ever to read a paper in a field, read the first one. Because what happens is in the first paper, you understand something. This paper, the authors now try to be more profound. And by the time we come to the 100th person in the field, the authors try to be so profound that nobody can understand what they're saying. And this is the paper by Lynn Marguler in Journal of Theoretical Biology. It wouldn't have been published in those days in any other journal. Very long paper. And this discusses the origin of mitochondria in a eukaryotic cell and how a eukaryotic cell might actually evolve. So actually now people believe that by fusion of a bacterium and an archaeon, because many of archaeal genes have commonalities with eukaryotic genes, other things like oxidative phosphorylation, etc., come from bacteria. The moment mitochondria were acquired, energy was now available, and the eukaryotic explosion then took place. If we go back to the timetable of evolution, which I showed you, just look at it again. It's somewhere around there, 2,500 million years ago, 2.5 billion years ago, that much of this dramatic things in the tree of life really happened. I brought this slide along because while I'm not a zoologist, I'm only interested as a layman in evolution. This paper appeared in 2019, and this is probably the evidence for the first animal that you might have. And of course, when you have animal biotechnology, you exclude many things, cows and buffaloes and things like this appear like animals. We too are animals. The others, there are lots of animals like this. You can see many things, segmented, bilateral symmetry, all of that. This one is about 500 million years. That's when things probably began in the field that you're interested. That book will ask what Nick Lane calls the vital question. And that is, why is life the way it is? Effectively, it's another way of asking, why is biochemistry the way it is? Biochemistry is so complicated that even those who study biochemistry don't want to study it. So in a biochemistry class, if you teach, every student has looked at the chapters on cellular metabolism. And every student has forgotten what it is immediately after the examination was over. Oh, a few names will be there, and that's about it. There appears no logic, there appears no connectivity, it doesn't appear to relate to anything. Now, the question is, what is most important for life? We will always think about DNA. I somehow have a prejudice against DNA. I shouldn't say this, you wouldn't like this. But I have a prejudice against DNA because even politicians know the word DNA. One will say corruption is in the DNA of my enemy, the other one will say autocracy is in the DNA of. They use the word DNA in a. What is more important in Lane's argument, more than information for life, eternity. And therefore, if you go back in time and back to this cell, you will find many things were already in place. ATP synthesis was already in place, and so was DNA. And so the competition really is between ATP synthesis and DNA as to what is most important. And there you can see oxidative phosphorylation as it is known today, extremely complex. And then I show you another article from the year 2000, article entitled, "Are You Serious, Dr. Mitchell?" And this was an essay written by the once inorganic and theoretical chemist, later term evolutionist at Francis Crick, Leslie Orgel. And what Orgel writes is, he says, chemiosmotic, fundamental and universal. This is Mitchell's chemiosmotic theory, where you have a proton gradient across the mitochondrial membrane, and it is proton gradient which drives ATP synthesis. One doesn't know the details yet, but they're still questions. But nevertheless, there is no other chemically reactive intermediate. Then he says, and this is what I liked very much, he says that biology does not have many counterintuitive theories. The last counterintuitive theory that biology had was Darwin. And that's why we find it so difficult even now to assess that. But he said, physics, on the other hand, many counterintuitive theories. Quantum mechanics completely counterintuitive. And who will like the idea of waves and particles, or cats which are both dead and alive at the same time, states getting entangled? It all makes no sense in real life. It's counterintuitive. He says the most counterintuitive theory says not since Darwin and Wallace has biology come up with an idea as counterintuitive as those of, say, Einstein, Heisenberg, and Schrodinger. And that he points to the chemiotic hypothesis for energy production of cells due to Mitchell. Mitchell still being argued. So in conclusion, because you are an Institute of Biotechnology, I will show you this. As I get old, I get really worried. Yesterday, I spoke to the students at CCMB. They wanted to know how things were long ago. So I was having this discussion with them. And as I was having this discussion, I realized that there were some words which had not entered my vocabulary 20 years ago. And what were those words? I hadn't heard the word innovation. I hadn't heard the word entrepreneurship. And I didn't know the word translation. I knew the word translation only when you translate something from one language to the other. I hadn't heard of bench outside and things like this. So these words have entered our vocabulary over the last 20, 25 years. Now, of course, if they have entered our vocabulary over the last 25 years, we must ask some questions about scientific research. There are two kinds of research. One is fundamental research. You want to find out what virus causes common cold, you look at it and find out what's done. The other is use-inspired research. Here I am, I'm going to solve the problem of malaria, I'm going to eliminate tuberculosis, I'm going to eliminate cancer, I'm going to do all those things. I don't even know the magnitude of the problem, but I'm going to solve all those. That is use-inspired research. Now, many years ago, the American political scientist actually wrote a book called Science, Technology, and Innovation. And he introduced this idea of what he called Pasteur's quadrant. Donald Stokes was his name. And Pasteur's quadrant is a space quadrant, a two-dimensional diagram. I put use-inspired research along the x-axis and fundamental research along the y-axis. So this is research space. If you want to do only fundamental research, you go vertically up, and there you have what he called Bohr quadrant. He died before his book was published, so it's just a square with a name. I put all the Google images into it so to make it a little bit more comfortable. There you have Bohr. Bohr's work was fundamental. Was it useful? Was understanding the spectrum of the hydrogen atom useful? Was the idea of a nucleus and electrons going around it, was it useful? Now, today, if I would tell this to a funding agency, they would say, no, don't try and explain the spectrum of the hydrogen atom. Nothing worthwhile, doesn't solve any of my major problems. But that's the Bohr quadrant. It's useful, nevertheless. Edison, on the other hand, could have discovered the electron. J.J. Thomson did. In Edison's notebook, there are notations which now tell you experiments that he was doing, which are getting to the same conclusion as the discovery of the electron. He wasn't interested. He wanted to illuminate your life. He was trying to make the bulb, and he produced the bulb. So that now is use-inspired research. Pasteur is the quadrant that everybody would aspire to, because in Pasteur's work, there were the most fundamental things. The resolution of tartaric acid set both organic and biochemistry completely ablaze, and finding out the causes of microorganisms as the cause of disease and other things, and everything else that followed. So that's the Pasteur quadrant. Now, where are we? The lower quadrant Stokes did not name. That's what I call average academic R&D. And I was now looking for an image to illustrate this quadrant. boar, pasture, and Edison. And the only image that came to my mind was Lakshman's common man. Now, the common man is always wandering around, trying to find a purpose. And so we are, in many ways, wandering around in research space, trying to find a purpose and asking the question, which direction should we go? Now, in any direction where you move, there will, of course, be obstacles. What would be kinetic barriers to crossing from one point to the other? The two-dimensional diagram is a very useful way of thinking about things. And of course, biotechnology has its roots in Avery's work on the pneumococcal transforming principle. If you read Avery's original paper, it was pneumococcal transforming principle. Nobody will read the paper again. He's not very sure also. He's a very cautious writer. And there he says that deoxyribonucleic acid might now be, and so on. He uses a lot of qualifying adjectives, very cautious man, which is why he didn't quite get the recognition that he deserves. It's sort of interesting. There are two dominant technologies. As I came, I found all the buildings in information technology. Bangalore, we actually have a minister for both information and biotechnology. So we think we're going to make money from all of this. Both of these came in the 1940s. They came from fundamental research. Claude Shannon's work, all the way, look at his PhD thesis title, An Algebra for Theoretical Genetics. Shannon's already thinking, genetics is information which is transferred. So genetic ideas are very useful in programming, in writing algorithms. And all of this was happening in the 40s. No one would have recognized these as useful at that time. So it is sometimes important to think about science and to think about research in a somewhat broader context, although it might be difficult. Lastly, I'd like to thank Dr. Taru Sharma for inviting me to give this lecture, to all of you for the very patient hearing that you've given me. And of course, to thank the two institutions where I've spent almost all my professional life and beyond. The Indian Institute of Science at the top left, where I was there for 41 years, and the National Center for Biological Sciences, the bottom right, which has given me a room in retirement. Thank you very much. I'm old enough for you to ask any question you like, even if it doesn't have any connection with the subject.
So, sir, in the last slide of yours, where you mentioned the average academic industrial R&D science. So, as a matter of fact, the government today, and in fact, DBD, as you said, is mostly interested in funding the translational research. So, we are a generation who are actually a part of the industrial revolution. This is made the core of the industrial revolution today. So, we are actually kind of being motivated towards translational research. Our heart still crave for the basic sciences. So, how do you think can we try to make the shift back? Because the government is not interested in funding mostly the basic sciences, as much they are interested in funding the translational research.
See, even translational research now, which the government is funding, I wouldn't blame the government as much as I blame scientists. The most difficult people are not government. Government only sends letters and memos and notices, and nobody ever reads them properly. But the committees which are formed of scientists have a very poor idea of what translational research means. Now, I've not come across anyone who's got a blockbuster patent, like the patents for PCR or the patents for cloning, which Boyer and Cohen had. We don't have those. At the same time, we have desire. The desire is that we should be useful. And if you are in the we must say that to get rid of disease. Not easy. Now, if you say that I'm not going to cure cancer, cancer is not one disease. Cancer is very complex. And even tuberculosis, which is caused by only one microorganism well studied, isn't going to be easy to get rid of. And as a result, we need to appreciate that even if we want to do translational research, we must have a very good fundamental understanding of what we are up against. And only then will we be able to translate well. Doesn't matter what the area is, whether it's new materials, new drugs, new ways of producing energy, whatever. You must have good ideas. How do you judge a good idea? Very difficult. I think one needs to be a little bit more forgiving of ideas that you don't like. Let them be done. So you can't let everybody thinks he has a good idea. So that you can't do. At the same time, you see the record of people. Now, if somebody comes to you with 15 patents, 15 patents for 15 publications. Before the open access revolution, you didn't have to pay for publishing. So if you had 15 publications, you had only spent money on research. If you had 15 patents, you also spent money on maintaining the patents. Now, for several years, if not a single patent has been licensed, then of course you must have some worry about putting more money into those. Just as if after 15 papers, if you find that nobody has read even one of those papers and cited it even once, you must have a little bit of a worry. So there are ways of judging performance of people. You can't judge a good idea. Very difficult to judge a good idea. But another problem with our institutions is protected intellectual property. I don't know whether intellectual property discussions happen here. They do. Now, my take on that, and I'm in a minority, so you don't have to use my word seriously. I wouldn't mind if somebody stole my idea. I'll tell you why. Because if someone stole my idea, that means independently someone has done it as a good idea. So if I got one good idea, then there's a good chance that I'll get another good idea. No? So I'll wait for a second. You see, surprisingly little research has been done on the 229E virus afterwards. Although in 2018, it was reported, there is one report of a patient who was infected with 229E and had what today we would call severe acute respiratory distress syndrome, SARS. Otherwise, 2019 was thought of to be only mild. But this is also because there's a vast difference in the responses of individuals to viral infection. If you ask yourself, although we say that COVID-2 was a terrible virus, The reason it spread was it wasn't a very terrible virus. The vast number of people who were infected were asymptomatic. And because of this, it spread in the world silently long before you actually recognize it had spread. Looking back in the early days of the pandemic, when there was this argument, ICMR's repeated statements that there has been no community spread of the virus were completely wrong. There's already been considerable community spread of the virus. It had spread in Italy. This is why it went so quickly from Italy to New York, because planes were going, everything was going around, it spread all over the world. And all of us did not react the same way. Susceptible populations were there. And then if you think about it a little bit more, no? This is an experiment done on us. You do it all the time with bacteria, isn't it? Put food and kill all the guys, the other guys remain resistant. So, to be truthful, I don't think I've ever had what people would call a eureka movement. In my reading, I don't think I've come across anybody who's had it. Archimedes was an exception. You take the alpha variant, you take the delta variant, you take the other variants, the beta variant, which is there in far, all of them have the D614 gene. They've accumulated more mutations.
Like for human infection,
This is the one, yes, absolutely. This would be the one, either way, whether it was created in laboratory or it was created naturally, there has to be a driver mutation. This is the driver mutation this process.
one more thoughts on this. But then we decide the fact that we have a number of genomes, we have proteins, which are even if they are making the same thing, they're still structured in very variable. But by that, nature has conserved the number of metabolites it turns out. The metabolites that biological system uses. And it's a huge what we call it, conservation across lines, from what the complex shows from the simplest line to the most complex line in terms of metabolite uses as well as the need. So these are the history of it. What do you know?
In fact, you see, it's actually interesting. I'm a chemist, and I shouldn't be saying, because I was trained originally as a chemist who made complex molecules. But today I would say, unlike in chemistry, laboratory chemistry, for a cell, it's much easier to make a protein or a nucleic acid than it is to make a metabolite. And I'll give you an example with which both bacteria and plants, microorganisms and plants, they make secondary metabolites. You call them secondary metabolites. But the secondary metabolites are very important for the plant or the bacterium. There's nothing secondary about secondary metabolites. How do you make secondary metabolites? You have a huge gene cluster, and those genes now make the enzymes. Look at the amount of energy that is needed now. You have to express all these genes, then you have to convert them into proteins, then all these proteins have to do the chemical reactions which transform one to the other. I don't have a slide here, but for some other talks I do have. You take something everybody likes. There's the universal cure for uh, everything. Uh-huh. That is curcumin. Uh, it's nice, it's local, it's our favorite molecule and everything. You want to make curcumin. Look at the number of steps that you be starting from two simple amino. You go all the way down to need, I don't know, 20 genes, 20 enzymes. So it's a huge investment of metabolic energy for the organism to biosynthesize a metabolite. So the metabolites are very hard to make, and therefore conserving the metabolites that once you've got, there's no particular reason to bury them. The starting points are also limited. Uh, you start with a carbohydrate starting point. Let's say you start with a 6 carbon sugar, put into glycolysis. There are only a few ways of going. You're never going to have, there's enough. I mean, the variation as it is bewildering. Oh, you mean no, no, I would guess microorganisms, you'll find more variety in secondary metabolites than in primary metabolism. Primary metabolism is the key to life. Secondary metabolism is the way in which the organism adapts to the environment in which it's found. Ah, you're in the current stage, if you're going to write a paper, you have certainly to work on your title, on your introduction, and on your conclusion, even if the results don't support your conclusion. Currently, what you're doing is you're not reporting your findings. You're actually selling your, you have to sell it to the editor, that's unfortunately what I would call a corporatization of science, where marketing is probably as important as anything else. I only say for all the younger people, tough luck. Can you use Aisha? Oh, for the tough luck. I don't really know. These things happen, and I'm sure, all organisms learn to adapt to their environment. And you will undoubtedly adapt. It's an evolutionary mechanism. You will adapt to whatever happens. I've seen that people now get grants because they promised things. But the only thing that you know when you sit and listen is they're never going to deliver on the promise. But the time it's the end of the grant, life goes on. I'm sorry to be cynical, but
Very much for joining us for this very fun. Yes, keeping all the talk that majority of the places. I think we would really go and put a vote of thanks. I thank also our
Thank you, ma'am. Thank you very much. And that was really an inspiring lecture. So on behalf of the event organizing committee, I'm glad to be standing here to express vote of thanks. I would like to thank our chief guest, Professor P. Balaram, for accepting our invitation for being here with us today on this special occasion and for enlightening our audience about reflection on science in the aftermath of the pandemic. I would also like to thank our director, ma'am, for her support, all the dignitaries which are present here and as well as online, all the scientists, staffs, and students for their presence. I would request all to join for a cup of tea outside the auditorium. Thank you very much. Yeah, before that, we can have